Welcome to the deep dive. You sent over some really interesting material on neuroepigenetics.
Yeah, it's a fascinating area.
It really is how our environment, our experiences can actually tweak how our genes function in the brain without, changing the DNA code itself.
Exactly.
So our goal today is to unpack that based on what you shared, maybe find some aha moments about how life literally shapes us.
Sounds good. The sources you sent clearly focus on that dynamic in the brain. It's less about the fixed blueprint.
Right, the DNA sequence we inherit.
Exactly. And more about the layers of control on top of that, how those genetic constructions actually play out in our nervous system.
Okay, let's get straight into it then. Based on those articles, what is neuroepigenetics? Maybe in simpler terms.
Well, it's the study of how things we experience, environmental signals, can flip gene switches in our brain cells.
On or off?
On or off, yeah. And the key thing your sources highlighted is that this happens without changing the underlying genetic code itself. That's the crucial distinction from, standard genetics.
And how do these experiences actually manage to do that? How do they trigger these switches in the brain, according to the research you looked at?
What really stands out from the materials is just how sensitive our brain cells are.
To everyday stuff.
Yeah, things like stress, diet, social interactions, even just learning something new, they can all send signals that lead to these epigenetic modifications.
So it's like making notes on the genes?
Kind of, yeah. Biological annotations telling specific genes when and where to be active or inactive.
Your notes mention a couple of specific mechanisms, DNA methylation and histone modification. Can you break those down briefly based on what you read?
Sure. So DNA methylation, your sources likely detail this. It's basically adding a chemical tag, a methyl group, directly onto the DNA molecule.
And that usually does what?
Often, it acts like a silencer, switches the gene off.
Okay. And histones.
Histone modification is different. It involves changes to the proteins, the histones that DNA wraps around like spools.
Ah, right.
These modifications can either loosen or tighten that wrapping.
Making the genes easier or harder to read.
Precisely. More or less accessible for activation.
Got it. Now, one thing that seemed prominent in the materials was early life stress. How does that epigenetically shape the developing brain?
Yeah, that's a big area. The research you shared probably emphasized that significant early adversity can cause pretty long-lasting epigenetic changes
Where in the brain?
in regions really critical for managing stress and emotions. And this can then influence, down the line, someone's vulnerability to mental health issues.
That sounds quite serious. But are these changes set in stone? Or can they be reversed based on what you found?
That's a really important question. And your source probably touched on this many epigenetic changes are actually reversible, or at least modifiable.
Oh, really?
Yes. While some modifications, especially early ones, can be quite stable, there's a lot of research now looking into therapies, environmental interventions,
Yeah.
things that might help sort of rewrite some of these patterns.
Which ties into the discussion in your articles about psychiatric disorders, I suppose. How does neuroepigenetics fit in there?
Exactly. Your readings likely pointed towards the idea that altered epigenetic patterns in certain brain circuits might contribute to conditions like, say, depression or anxiety.
How so?
Well, changes in gene expression driven by these epigenetic marks can disrupt the normal functioning of those circuits.
But it's not all about problems, is it? The sources mention learning and memory, too. Does epigenetics help us there?
Oh, absolutely. Your materials probably highlighted that epigenetic modifications are thought to be, well, essential for the brain's plasticity, for forming new memories, for learning.
So they help strengthen connections.
That's the idea. These
Oh.
changes help strengthen synaptic connections, solidify new information. It's part of the learning machinery.
Okay. And what about the idea of passing these changes down generations? That sounds almost sci-fi. Did your sources cover that?
Yes, the concept of epigenetic inheritance. It was likely mentioned.
Air.
Well, there is some evidence, mostly from animal studies currently, suggesting that some epigenetic changes in parents might be transmitted to offspring.
Potentially influencing their development, too.
Potentially, yes. However, your sources probably also noted that how much this happens in humans and the mechanisms are still very much under investigation. It's complex.
I can imagine. Studying this in the living human brain must be incredibly difficult. What challenges did the readings point out?
A major one, as your sources likely mentioned, is just accessing the tissue. You can't easily take biopsy samples from a living person's brain for epigenetic analysis.
Right, so you rely on models.
A lot of research uses animal models or studies post-mortem human brain tissue. Plus, the brain itself is so complex, countless different cell types. each potentially with its own epigenetic profile. It adds layers of difficulty.
But if we can get past those hurdles, what's the potential payoff? Especially thinking about personalized medicine based on what you read.
The potential is huge, really exciting. Your sources may have suggested that understanding a person's unique epigenetic signature could lead to much more tailored treatment.
For things like depression or Alzheimer's.
For neurological and psychiatric conditions, yes. Maybe even developing therapies that directly target specific epigenetic modifications to correct them.
So, okay, pulling this all together, the big takeaway from your sources really seems to be that our lives, our experiences, leave a tangible biological mark.
Definitely.
Yeah.
They have a real impact on how our brains function through these epigenetic mechanisms,
Our lives are constantly interacting with our genes in the brain.
shaping who we are in really fundamental ways. It's not just nature or nurture, it's the interplay.
It really makes you think, doesn't it? Especially considering how early life experiences can have these lasting epigenetic consequences. What are the broader societal implications, for how we support healthy development across the entire lifespan?
That's a crucial question to ponder.
Definitely food for thought. Thanks for digging into this with us today.